Biosilk is a completely defined biomaterial made of recombinant silk protein that can be easily functionalized with biolamina's biolaminin substrates. This video describes how to generate Biosilk 3D foam scaffolds for the expansion and differentiation of human pluripotent stem cells. The cell suspension has to be concentrated so that the volume ratio to the Biosilk solution does not exceed 1:5. Also make sure that it is prepared freshly before mixing into the Biosilk solution to ensure the best cell viability. Thaw the Biosilk solution at room temperature. Do not vortex or shake the Biosilk vial. It will take around 12 minutes from the frozen Biosilk solution to thaw at ambient temperature. The thawed Biosilk solution will gradually turn milky in ambient room temperature. For best result, the thawed solution should be used as soon as possible. Latest within 1 hour of removal from -80 degrees Celsius. To make Biosilk foams, prepare pipettes for volumes ranging from 1 microliter to 1,000 microliters and 24-well hydrophobic surface plate. For the 3D scaffold formation step, there are 2 options to make the Biosilk foam. The first option is the one-step approach, which is suitable for less sensitive cells. To the thawed Biosilk solution, gently mix together the biolaminin of choice, cell suspension, and ROCK inhibitor. Transfer 22 microliters of the mixture to the center of 1 culture well. Pipette quickly air bubbles into the droplet using a pipette set at 44 microliters to obtain a 3D foam. Spread out the foam in circular motions with the pipette tip. Repeat the procedure to create the desired number of foams. 1 vial of Biosilk is sufficient for 12 foams. The second option is the sequential approach, which is suitable for sensitive cells. Prepare a mixture of the thawed Biosilk solution with biolaminin of choice. Add ROCK inhibitor to a final concentration of 10 micromolar. Transfer 20 microliters of the prepared Biosilk biolaminin solution to the center of 1 of the culture wells. Use a pipette set at 44 microliters and push air bubbles into the droplet by quickly pipetting up and down 22 strokes to obtain a 3D foam. Spread out the 3D foam in circular motions with the pipette tip. Immediately, add 1 to 4 microliters of the cell suspension to the 3D foam. Use the pipette set at 40 microliters with a new tip and disperse the cells throughout the 3D structure by 5 additional strokes. Repeat this procedure to create the desired number of 3D foams. 1 vial of Biosilk is sufficient for 12 3D foams. Now, place the plate with the cell-containing 3D foams in an incubator at 37 degrees Celsius for 20 minutes. During this time, the Biosilk biomaterial polymerizes and the 3D structure is stabilized. Remove the plate from the cell incubator. For each well, add 700 microliters to 1 milliliter of pre-warmed medium containing 10 micromolar ROCK inhibitor. Start dropwise around the 3D foam and slowly fill up to cover the whole 3D foam. Place the plate back into the incubator. Feed the cells daily or at appropriate frequency with fresh culture media without ROCK inhibitor. After the Biosilk 3D foam formation, at day 1 it is expected to see the small bubbles starting to merge into bigger bubbles. At 72 hours, larger bubbles will disperse, resulting in a 3D scaffold with cells integrated between the microfibers within 4 days. In this last section, we will look at the preventative measures that should be considered to avoid foam dispersion or floating after medium addition or after 24 hours of Biosilk 3D foam formation. In order to succeed with your Biosilk 3D culture system, we encourage you to read these 9 tips carefully.